My name is Rachel Haurwitz, and I'm the president and CEO of Caribou Bio. Caribou is a CRISPR genome editing company spun out of Jennifer Doudna's lab at the University of California, Berkeley. We have developed a next-generation CRISPR technology that is dramatically more specific than the first generation of CRISPR-Cas9. It allows us to safely make multiple edits to a population of T cells. And using this technology, we are developing a wholly owned pipeline of off-the-shelf gene-edited cell therapies for cancer. Through our multiplex modifications, we are able to not only make the edits necessary to ensure that we do not cause graft versus host disease with our allogeneic cell therapies, but we can further directly boost and armor the T cells for better anti-tumor activity. As we think about the role of cell therapy in the future of cancer development, we at Caribou believe that the future of cell therapy will be allogeneic or off-the-shelf. Allogeneic cell therapies hold tremendous potential to take advantage of the initial clinical responses achieved with autologous cell therapies, while overcoming so many of the challenges presented by a patient-specific approach to cell therapy. However, there is an important challenge to allogeneic cell therapies that we believe can be overcome using our genome editing capabilities, and that is specifically T cell persistence. Allogeneic cell therapies are fundamentally foreign cells. They come from a healthy donor, are manufactured into a product, and delivered to a cancer patient. Eventually, the cancer patient's immune system will recognize these as foreign and clear the therapy. And this is very different from the autologous setting, where a single dose of cell therapy might persist for many years after a single delivery. And so at Caribou, we use our next generation genome editing capabilities in order to directly boost T cell persistence. And we think about this in multiple ways. In our lead program, we directly address the issue of T cell exhaustion, and we do so by genetically deleting PD-1 from the CAR T genome. And as you'll see today, this deletion of PD-1 leads to a dramatic increase in the durability of anti-tumor activity in vivo. We can additionally approach persistence by immune cloaking the CAR T cells. And we do that in our second program by getting rid of endogenous HLA class I presentation, and in its place, engineering the cells to only express HLA-E, one of the minor antigens. This will keep both the patient's T cells and their natural killer cells at bay, slowing down immune-mediated clearance and buying additional time for additional anti-tumor activity. Caribou's wholly owned pipeline consists of multiple allogeneic CAR Ts, as well as a natural killer cell platform for solid tumor targeting. Our lead program, CB10, is an allogeneic anti-CD19 CAR T. We've wrapped up the IND-enabling studies and anticipate launching the phase 1 clinical trial for this program before the end of this year. I'll tell you more details about this, including some of the animal data, in just a moment. Our second program, CB11, is an allogeneic anti-BCMA CAR T, and it is set to enter the clinic next year. And this is where we use our immune cloaking strategy in order to boost the allogeneic CAR Ts. Behind that, we have other areas of discovery research, including further allogeneic CAR T programs, as well as a cell therapy effort to target solid tumors, centered on editing induced pluripotent stem cells in a variety of ways, such as arming them with a CAR, or further armoring them with other capabilities to boost their proliferation and other anti-tumor activity capabilities, and then differentiating those into natural killer cells that could be targeted to a variety of different solid tumor types. At Caribou, our next generation CRISPR technology platform relies on new guides that have been developed by some of my colleagues that dramatically improve the specificity of targeting either Cas9 or Cas12a to specific sites in the genome. Of course, both of these proteins come from natural systems where they're guided by all RNA guides to the right site in the genome. What we have done instead at Caribou is develop hybrid molecules that have some DNA nucleotides as well as RNA nucleotides. We call these the CRISPR hybrid RNA DNA, or chRDNA for short. and it's the inclusion of DNA that dramatically improves their specificity. We have a strong IP position behind these technologies, including 6 issued US patents claiming Cas9 and Cas12a chRDNAs. Caribou has the exclusive rights to deploy Cas9 chRDNAs for CB10, and Caribou has broad exclusive rights to use Cas12a chRDNAs for all therapeutic purposes. Both technologies, both Cas9 chRDNAs and Cas12a chRDNAs, lead to very high efficient genome editing with no detectable off-target effects in human primary T cells. And examples of those data are shown here on both the left and the right side of the slide. If you start looking on the left side of the slide first, this is a head-to-head comparison looking at the first generation all RNA guides with Cas9 versus the next generation chRDNA guides. And what we see is that every time we use these first generation all RNA guides, we observe at least one, if not multiple, off-target effects. But when we shift and target the exact same site with chRDNAs instead, we observe no off-target editing down to our limit of detection. And if you look at the right side of the slide, it's a very similar story for Cas12a. When we use Cas12a chRDNAs, we observe no detectable off-target edits down to our limit of detection. So this is really critical as we think about ensuring that we make only the edits that we intend to our T cell genomes and avoid off-target consequences elsewhere. But the specificity of a given reagent is only one piece of the puzzle as we think about the genomic integrity of these cells. And the other comes down to preventing chromosomal rearrangements or other translocations. This can happen whenever a cell experiences 2 simultaneous double-strand breaks, which can occur at, say, both an on-target and an off-target location. And so by dramatically reducing the frequency of off-target editing, we reduce the risk of those off-target translocations from occurring. However, what I've shared with you is that Caribou is intentionally making multiple edits in our T cell programs. And therefore, we have to face head-on the challenge of preventing translocations between the multiple sites that we intend to modify. And so at Caribou, we've developed a proprietary delivery technology that significantly reduces the frequency of these translocations. And I'm showing an example of that here. On the left is a schematic of what could occur. In, for example, our lead program, we edit both the TRAC and the PD-CD1 genes. And therefore, the T cells could create a translocation between these 2 chromosomes. And in fact, if we use a standard delivery approach shown in the middle box, that's exactly what we see. In this case, about 3.5% of the T cells have translocations between these 2 edited genes. And so by using our proprietary delivery technology instead, we're able to dramatically reduce this frequency down, in this case, to 0.1%. So between the stringency of the chRDNA reagents themselves and this delivery technology, we are able to safely and carefully deploy multiplex genome editing in our CAR-T development. We use these technologies to develop our wholly owned pipeline. And as I mentioned, CB10 is Caribou's allogeneic anti-CD19 CAR-T manufactured using the Cas9 chRDNA technology. This is where we remove the T cell receptor to prevent GVHD. We site-specifically insert the CAR, and we further delete PD-1 in order to boost T cell persistence. These data here demonstrate the utility of a PD-1 deletion and why we believe CB10 with PD-1 deleted holds the potential for being the best-in-class anti-CD19 allogeneic CAR-T. What you're looking at here is an in vivo head-to-head analysis of CB10 compared to a conventional allo anti-CD19 CAR-T. In both of these CAR-Ts, we've used our next generation genome editing to remove the T cell receptor and site-specifically insert the CAR. So the only difference between these 2 products is that in the conventional CAR-T, PD-1 remains on, whereas in CB10, PD-1 has been deleted from the CAR-T genome. We deliver a single dose of each of these into mice experiencing metastatic disease and watch and wait. And what we see is that initially, both are quite efficacious. And by 14 days after delivery of a single dose, none of the treated animals have any detectable tumor burden remaining. However, what happens next is completely different. the animals treated with the conventional product see their tumors recur fairly quickly. And by day 108, half of those animals have expired from their recurrent tumor burden, and the half that remain are back to full-blown metastatic disease. By day 130, all of those animals have expired. In strong contrast, the animals treated with CB10 are not only still alive by day 108, half of them still have no detectable tumor burden. And those that are starting to see their tumors recur have only a very mild amount of disease, certainly nothing like the metastatic disease experienced by the other cohort. And through day 160 post a single dose, all but one of the CB10 treated animals are still alive. So these data really demonstrate to us the power of deleting PD-1 from a CAR-T genome and give us the confidence to believe that CB10 holds tremendous potential for patients with non-Hodgkin lymphoma. Manufacturing, of course, is critical to an allogeneic CAR-T cell therapy, and we've invested heavily in process development in Caribou, where our PD team is responsible for developing the manufacturing process, scaling it up, and tech transferring it to our CMO partner, who then follows our steps to make the gene-edited CAR-Ts. The CMO not only makes the CAR-Ts, they go through a final step that we've developed to selectively remove any residual TCR-positive cells, leaving vanishingly few cells in the final cell dose. We anticipate launching the ANTLER Phase 1 clinical trial for CB10 before the end of this year. This will be a trial targeting patients with relapsed refractory non-Hodgkin lymphoma, and we anticipate enrolling up to 7 different subtypes of this disease. In the first part of this trial, we'll be going through a very traditional 3+3 dose escalation in order to evaluate safety and tolerability and to identify the recommended Phase 2 dose. Then in a second part of this Phase 1 trial, we'll carry out an expansion phase in 1 or more of these subtypes in order to collect initial efficacy data at the recommended Phase 2 dose. We very much look forward to the opportunity to evaluate the safety and ultimately the efficacy of CB10 in patients beginning later this year. A few words about CB11, Caribou's second program. This is an allogeneic anti-BCMA CAR-T that we manufacture using our Cas12a chRDNAs. Here we not only get rid of the T-cell receptor and insert the CAR, but we further manipulate HLA class 1 presentation so that most of the endogenous antigens are no longer expressed and only an engineered HLA-E is expressed to prevent both the patient's T cells and their natural killer cells from clearing the therapy. To the best of our knowledge, Caribou will be the first group to deploy this type of technology in the clinic, and we very much look forward to bringing this program into the clinic next year. This is very challenging from an engineering perspective to develop, as it requires 2 deletions and 2 separate site-specific insertions. And we have found that only Cas12a chRDNAs mediate sufficiently high site-specific insertion to carry out the manufacturing process to result in a high number of T cells that have all of these different modifications at the end of the process. And finally, not only does the immune cloaking strategy set CB11 apart, but so does the CAR. Caribou has acquired a new humanized anti-BCMA CAR that is far more potent than any other anti-BCMA CAR that we have evaluated. So between the CAR and the immune cloaking strategy, we believe CB11 holds tremendous potential for being the best-in-class anti-BCMA allogeneic CAR-T. And finally, just to remind you, we additionally have work going on at Caribou evaluating the potential of editing iPSCs, differentiating them into natural killer cells, and targeting them to a variety of different solid tumor types for additional cancer cell therapies. And with that, thank you very much for your time and attention. And I welcome any reach out from those of you in the audience. Thank you.